Professor Nick Lane from UCL, United Kingdom. Sir, welcome to today's event. His topic would be the energy and matter at the origin of life, and allow me to say a few words as an introduction. Professor Nick Lane is a professor in evolutionary biochemistry at UCL. He graduated from Imperial College London and thereafter earned his PhD in 1995 from Royal Free Hospital Medical School. His research interests includes evolutionary biochemistry and bioenergetics, focusing on the origin of life and the evolution of complex cells. He has published more than 80 peer-reviewed papers in top international journals, including many in Nature and Science. He played a crucial role in the establishment of the UCL Consortium for Mitochondrial Research and the UCL Center for Life's Origin and Evolution. He has authored 4 popular books on evolutionary biochemistry, and you might have seen them: The Vital Question, Oxygen, The Molecule That Made the World, right, among others. He has gained significant international recognition, including the Royal Society Michael Faraday Prize in 2016, the Biochemical Society Award in 2015, and the BMC Research Award in 2011. He's a fellow of the Linnean Society, the Royal Society of Biology, and the Biochemical Society. So without further ado, it gives me great pleasure to invite Professor Nick Lane to make his presentation, sir.
Thank you very much. Let me just try and share my screen, if I may. Can you see that?
Yes,
yes sir.
Yeah.
Yes,
thank you.
Great. Well, thank you very much for the invitation and the kind introduction. I will give a presentation for about 45 minutes or so, and then happy to take questions after that. I hope that's a reasonable balance. If you would like me to cut the presentation short, please just let me know. But I would like to give a general perspective on the origin of life from a biochemist's point of view. I know that Schrödinger is a popular figure. His famous book in 1944 entitled What is Life really asked 2 questions as far as I'm concerned. One of them linked to genes and information. And he was the first person really writing about biology to refer to the term code script. So this was during the Second World War. It was a period of when encryption was important, and a lot of physicists were interested in information, the theory of information. And so this phrasing is quite modern. He used terms like code script in thinking about the genetic code, and predicted that there should be an aperiodic crystal, which is effectively what DNA is. And so he had quite a lot of foresight as far as information is concerned. He also talked about entropy, and there I find his terms more archaic and old-fashioned. He talked about life feeding on negative entropy and continually sucking orderliness from its environment. And I don't really know what he had in mind when he talked about that. These are not terms that mean much to biochemists, but he did say in a footnote to the 2nd edition, if I had been catering for physicists alone, I should have let the discussion turn on free energy instead. Now, it's important to say that when Schrödinger was writing in 1944, this was 10 years before Watson and Crick and the structure of DNA as a double helix. But it was more than 20 years before the discovery of free energy transduction in biology, which was really in the 1960s when this first came to be understood. This is work by Peter Mitchell and a lot of the experimental work done by Jennifer Moyle. And this is Mitchell and Moyle in the late 1940s in Cambridge. And the picture that I have here is taken from a Mitchell paper in 1967, which was really the first time that he had presented something that looks modern to us in terms of how respiration and energy transduction in biology actually works. This was radical. There's a very nice quote from Leslie Orgel, who said, "Not since Darwin has biology come up with an idea as counterintuitive as those of the great physicists, Einstein, Heisenberg, or Schrödinger. And people were thinking about the cell really as a bag of chemicals. And what Mitchell was saying is that it's the bag itself which is the important part. It's the membrane surrounding cells where all this activity is taking place. And it's electrical activity. So the energy from burning food in oxygen in respiration powers the extrusion of protons across a membrane to generate what he called the proton motive force, which is to say there is an electrical charge on the membrane because protons are pumped outside the cell in a bacterial cell. And this proton motive force drives the synthesis of ATP. So this is how respiration works at the cellular level. And since this original conception in 1960s, we have learned, biochemists have learned a tremendous amount of detail about how respiration actually works with atomic level of resolution for some of these complexes. So this is a modern respiratory chain that I'm showing you now. So at the left-hand side is complex 1. There are multiple complexes, 4 complexes, which are pumping protons across the mitochondrial membrane. So this is happening inside our cells, inside our mitochondria. And this is what's happening when we're respiring food. We are generating an electrical charge on these membranes. The electrical charge and the flow of protons is powering ATP synthesis. ATP is usually called the universal energy currency of life. So it's an extraordinary system, this. The charge on this membrane is in the region of 150 to 200 millivolts, which if you were to shrink yourself down to the size of a molecule is equivalent to 30 million volts per meter as a field strength. So that's equivalent to a bolt of lightning. So the charge on these membranes, which are very thin, 6 nanometers thick, is an enormous electrical potential across a very short distance. This work led on to a Nobel Prize for Sir John Walker and Paul Boyer in 1997 for working out the structure of the ATP synthase, which works more or less like this. It's a rotating nanomotor. It's an extraordinary molecular machine. In some sense, all enzymes are molecular machines, but this is such a beautiful example of just how natural selection can hone molecular machinery to really function in a way that we would recognize as a motor in this particular case. And other pumps as well. So this is complex 1. The structure of this in mammals was worked out a few years ago by Judy Hurst. This is a kind of an ongoing process. The level of resolution gets smaller and smaller. This is much more complicated than the bacterial versions that have been that have been worked out the structure of before. And again, we now know in pretty much atomic resolution how proton pumping is working at the level of these proteins. So it's an extraordinary level of detail. But we have drifted a long way from the questions that were of interest to Peter Mitchell and Jennifer Moyle in the first place. So this is a quote that I like a lot from Peter Mitchell at a conference on the origin of life in Moscow in 1957. And he wrote, "I cannot consider the organism without its environment. From a formal point of view, the two may be regarded as equivalent phases between which dynamic contact is maintained by the membranes that separate and link them." So when he's talking about organisms here, he's not thinking about human beings or multicellular organisms. He's thinking about bacteria. And he's essentially saying that bacteria are surrounded by a cell membrane. They're enveloped by a cell membrane, which is very thin. As I say, it's 6 nanometers thick. And remarkably, he's talking about equivalent phases between the inside and the outside of this membrane. Now, it's hard to think of life as being an equivalent phase to the external environment. It's a radical thing to say, but if you're thinking about the origin of life, before there is such a thing as a cell that we would recognize, then this is actually a very helpful way of seeing it, because we're talking about barriers and we're talking about different conditions on opposite sides of barriers. So the question that Mitchell was interested in, especially at the beginning, was how is it that bacteria keep themselves, their insides, different to the outside world? These two phases across this membrane, how do they maintain a difference in the composition between the inside and the outside? How do bacteria keep their pH stable? How do they keep their ionic balance stable? How do they keep molecules in, import food if they need food, import and excrete waste? And he realized that it had to be done with the same level of molecular recognition as an enzyme, that proteins that sit in the cell membrane, and the example I'm giving here is a proton, need to be recognized and pumped, physically pumped out of the cell. And plainly, that costs energy to do that. And what he realized was that if you then allow them to flow in again, if you allow, and all pumps in biology will work in either direction, if you allow a proton gradient to flow in, then in principle, it can power work. And that work in respiration is ATP synthesis. And so he made this link between how bacteria keep their insides different to their outside and how respiration works in human beings. And that was the intuitive leap that Mitchell made. Now, it is generally true that bacteria are powered, if you like, by letting the outside in. And the outside in this sense is rather specifically ions like protons or sodium ions and so on. And they are pumping these out and allowing them to flow in. They're pumping out using energy from respiration, from either reactions between minerals or burning food. And the inflow of protons powers growth. It's powering both carbon and energy metabolism. And this is why I entitled my talk Energy and Matter, because converting gases into organic molecules is powering growth more than anything else. And of course, it costs energy to pump anything outside again. And these pumps, as I've shown you, are enormously complex structures. It is very difficult to recognize a proton is essentially the smallest meaningful particle in biology, apart from electrons, of course, and it's being recognized and pumped out. That's an extraordinary thing to do. It's not something that we can assume would happen very easily in the absence of genes and information. And so how did it arise? And it's universally conserved across all of life, implying that there's something fundamental about cell structure in how this works. And so the question is, well, what came first? Did these pumps arise very early? Or is it possible that there's a geological environment which is doing the same thing and powering the origin of life? And that's what I'd like to put to you today. So how can we possibly know? Well, phylogenetics, phylogenomics over the last 20 years or more has given a very radical view of the tree of life, quite different to what was understood previously. So we are eukaryotes, we're complex cells with a nucleus and plenty of DNA and lots of internal structures. But eukaryotes are a derived domain. They're derived from a symbiosis, an endosymbiosis between bacteria and archaea. And so we cannot trace our own roots directly back to the last common ancestor, last universal common ancestor of all of life. So this is a tree from 20 years ago, a conceptual tree done by Bill Martin, who has been one of the leading thinkers in this field over several decades now. And the other thing I'd like you to notice here, this common ancestor of all of life, Luca, is the common ancestor of the bacteria and the archaea, which I'm showing here. They're rather simple cells in terms of their morphology. They lack the complexity of eukaryotes. And in this, Bill is showing two separate emergences from a common ancestor that lived inside a hydrothermal vent. And that was a radical proposition, and indeed still is a radical proposition. The reasons for it are quite simple. There are fundamental differences in cell structure, in biochemistry, and in genetics between bacteria and archaea. They plainly do share a common ancestor because there are many things that bacteria and archaea have in common, including the universal genetic code, the structure of ribosomes. These are the protein building factories inside cells. The genes and therefore the enzymes involved in transcription, which is to say reading off DNA into RNA, and translation, which is to say converting RNA sequence into a protein, a sequence of amino acids in a protein. Parts, but not all, of intermediary metabolism. That's to say the core metabolism of cells is universally conserved, but the genes that are involved in catalyzing these pathways are not universally conserved. Not even the Krebs cycle or glycolysis are universally conserved in their genes. And broadly, membrane bioenergetics are conserved. Both bacteria and archaea are using a charge on a membrane to drive growth. But the things that are different is the most important one for my talk is the cell membrane itself is different in its chemistry in bacteria and archaea, and not just the chemistry, but then the enzymes required to make the lipids in the first place that make up these membranes. The cell wall is different again in its chemistry. Pathways like glycolysis, which many people think of as ancient, differ in the genes in the bacteria and archaea. They're not homologous genes. Even DNA replication is not homologous in bacteria and archaea. The genes and the proteins involved in replicating DNA are not the same. There's only a handful of them which are similar. Um, and respiratory chains, these are the pumps themselves. There's been a lot of lateral gene transfer, so genes can be passed around between bacteria and archaea, almost like loose change. And that confounds trying to draw a tree of life for them, but it looks as if there is a fundamental difference. So there are some really paradoxical differences. And the real paradox to me, I'm going to try and encapsulate it in a nutshell, is membrane bioenergetics are universal, but the membranes themselves are not. And this paradox could be resolved if life originated in an environment where there were natural, geologically sustained proton gradients that were doing the same job that pumping to generate a biological proton gradient is doing today in cells. So is there such a place? The answer is yes. It was discovered 20 years ago by Deb Kelly, who was captain of the Alvin submersible. And this is 20 miles or so away from the mid-Atlantic ridge. This is a vent field known as Lost City. And these are unusual vents. These are alkaline hydrothermal vents. They're not like black smokers. You can see she's holding a lump of a black smoker here, plus a piece of Lost City. They're quite different in their structure and in their chemistry. The two things to appreciate is they're called alkaline because these alkaline fluids, the hydrothermal fluids, are strongly alkaline, pH 11 very often. And they're rich, they're bubbling in hydrogen gas. They're produced by a reaction in the crust called serpentinization, which is essentially water reacting with minerals like olivine to metamorphose these minerals. And what is released are hydrothermal fluids, strongly alkaline and rich in hydrogen gas. So these bubble back up to the surface where they precipitate as these hydrothermal vent fields at the bottom of the oceans. This kind of vent was predicted 20 years before they were first, 10 years before they were first discovered by Mike Russell. His ideas in the 1990s were important, but peripheral, I would say, to the field. And it wasn't until the discovery of Lost City that his thinking about the way that life might have started became mainstream. And this was a Photoshop image of Mike Russell done by Nature in a feature article in Nature in 2006, where they dressed him up as Renaissance man, as Erasmus, and called him Naissance man, naissance as in the birth of life. And there's a little bit of Lost City vent behind and the reactor that he built at the Jet Propulsion Laboratory. And this is showing at the right-hand side here is showing the structure of Lost City, a little piece of Lost City. And what you can see is it's like a mineralized sponge. This is a labyrinth of pores, interconnected pores, and the hydrothermal fluids will percolate their way through these pores. And so these are really electrochemical flow reactors. This was Mike's phrase, in which we have high concentrations of hydrogen in the hydrothermal fluids, high concentrations of CO2 in ocean waters that percolate into these vent systems, and strongly alkaline fluids with mildly acidic ocean waters generating quite steep pH gradients, proton gradients across what could have been catalytic iron sulfide, iron nickel sulfide walls. There would have been other things in these walls as well, quite different in structure to modern vents where the oxygen has changed the chemistry of the oceans. But in the Hadean 4 billion years ago, we think there would have been much more iron, ferrous and ferric iron, and nickel and sulfides in these walls, as well as silicates and more conventional minerals. This is what got me interested in this field. There is a similarity in both the magnitude and the polarity of these hydrothermal vent pores and a bacterial cell. So this is the image I showed you before of a bacterial cell with a membrane pump pumping protons out. So this is how we respire. This is also how bacteria respire. They pump protons out. Um, and so there's a difference in proton concentration between the outside and the inside of the cell of about 3 pH units, equivalent to 3 pH units. So a 1,000-fold difference in concentration. And we should see something very similar here, hydrothermal fluids, alkaline fluids, with a barrier containing iron sulfide minerals, and outside relatively acidic ocean waters percolating into the vent system. So potentially a pH gradient of 3 or 4 pH units across this barrier as well. So there's a similarity in structure here. And the question is, can this inorganic structure power work in a prebiotic setting? Can it power something like CO2 fixation? The reason to think it might is that the iron sulfur minerals that I've been talking about, things like greigite and McKinnowite, which you can see the structure at the left-hand side here, we find very similar structures in the proteins in the enzymes that are involved in CO2 fixation today. So ferredoxin, for example, contains what are called iron sulfur clusters. And there are various types of iron sulfur cluster, and they share the same mineral structure as the greigite half cell. So it's possible to imagine a halfway house, if you like, between a mineral environment, a hydrothermal environment, and modern cells, where we have something equivalent to a protocell, which is using these iron sulfur clusters to reduce CO2 and power growth. So is there any way of getting at this question? We do know that ferredoxin is ancient. This was a beautiful paper from Margaret Oakley Dayhoff in 1966, where she had deciphered the structure of ferredoxin and argued that this was an ancient protein, that it derived from doubling of even shorter proteins, and a repeating sequence of some of the most ancient amino acids. And she suggested that it meant that these were incorporated into metabolism very early during biochemical evolution, even before the complete modern genetic code existed. This was from 1966, so a remarkably prescient view. We certainly think of ferredoxin and related iron sulfur proteins as being ancient in that sense. So the acetyl-CoA pathway is reacting. This is a modern pathway in bacteria and archaea, and it's reacting hydrogen and CO2, exactly the same substrates that we have in these vent systems, to produce methane or acetate. This is the only pathway of CO2 fixation that's known in both bacteria and archaea. So in methanogens, which are archaea, and acetogens, which are bacteria. And that implies it was present in their common ancestor. Is overall exothermic. In other words, the reaction between hydrogen and CO2 releases energy, which in principle can be captured and used to power work. And we see this requirement for hydrogen and CO2 at almost every step in core metabolism of modern cells. And so this is a schematic showing, all I really want you to notice from this is we're starting with CO2 here, activated CO2, and in brown you see hydrogen, 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 carbon, more hydrogen here, more CO2, more hydrogen. Almost every step we're dealing with CO2 fixation and hydrogenation to produce from CO2 itself molecules now like acetyl-CoA, but there are prebiotic equivalents like methyl thioacetate, for example, into Krebs cycle intermediates, pyruvate, oxaloacetate, alpha-ketoglutarate, and so on, into sugars from pyruvate to glyceraldehyde, ribose phosphate, and so on. So this is the core, the deeply conserved core of biochemistry, and you can see it's all about hydrogen and CO2. And we get to these amino acids like aspartate, glutamine, glycine, for example, an energy currency, acetyl phosphate. In red here, I'm showing you these are the precursors for nucleotide synthesis. So to make the building blocks of RNA and DNA, this is what biology starts from. And you can see they're very close to the Krebs cycle intermediates, which are formed directly and have been formed in experimental work from hydrogen and CO2. So the blue arrows, I should say here, are showing reactions that have been accomplished under prebiotic conditions in the lab. not necessarily always under the same conditions and not by the same group all the time, but most of these reactions have in fact already been done now. And in some sense, it should not be surprising because overall, total cell biomass is favored thermodynamically. This is work from Jan Ammond and Tom McCollum. What you're seeing here is theoretical thermodynamic modeling under hydrothermal conditions, under alkaline hydrothermal conditions, so pH 11 and temperatures between 25 degrees centigrade and 125 degrees centigrade. And what you can see is that the free energy change is negative, which means to say it's exothermic, it's releasing energy when they react to form total cell biomass. And most of that energy contribution is from amino acids, also some fatty acids. It costs energy to make nucleotides. But if you can conserve some of the energy from down here and put it into making nucleotides, then it's possible to make those. And you can see if you, in principle, from a thermodynamic point of view, under these conditions, if you have hydrogen and CO2 in a test tube under those conditions, shake it, they should react to form cells. It is the favored end product is to form cells, not the other way around under those conditions. But of course, it doesn't happen. It doesn't happen because hydrogen and CO2 are stable molecules and do not easily react. And you can see that here, that the first couple of steps up to formaldehyde in a geochemical environment, a hydrothermal type environment, are thermodynamically uphill. It costs energy to get to formaldehyde. After that, it's downhill all the way. And what cells do is use a proton gradient across the membrane. We're back to membrane bioenergetics here to lower this barrier, this thermodynamic barrier to reaction. So this is showing the free energy change, and it's uphill to get through the first couple of steps, and then it's downhill to methane or acetyl. And in red, in this region here, this is the redox state of most things inside living cells today. That's the kind of the range. So there's not very much energy difference between CO2 itself and the state inside cells, the redox state of organic matter inside cells today. Why is there a barrier there? The reason relates to pH, and this barrier can be abolished by modulation of pH. So what you can see on the left-hand side here is that hydrogen does not have the reduction potential to pass its electrons onto CO2. It's energetically uphill. Hydrogen can't force its electrons onto CO2 to make formaldehyde. It's at pH 7 or any other pH, it's energetically uphill. So it will not happen spontaneously. But in these hydrothermal vents, we have hydrogen gas at pH 11. And that makes it more reactive. I can explain why if people are interested later on, but essentially if you're pushing your electrons onto CO2 in an alkaline environment, what's left behind is protons. They will react with hydroxide ions very readily and form water. And so this is strongly thermodynamically favored. So it will happen more readily in an alkaline environment. That means hydrogen becomes more reactive in an alkaline environment. And CO2, the opposite way around, CO2 becomes more easily reduced. It will pick up those electrons more readily in an acidic environment, as in the early oceans, because it picks up an electron and then it picks up a proton to balance the charges. Then it can pick up another electron and then another proton and so on. So it becomes easier to reduce in acidic conditions. So the conditions we have in vents should promote the formation of organic molecules from CO2 and hydrogen, but they have to be in separate phases. If you have mixing, then it will not work anymore. As soon as you have mixing, we're back to this phase. So we require barriers that prevent mixing. Now, we know that the most ancient cells, like methanogens, for example, what they're doing when they're reacting hydrogen with CO2 is a process called electron bifurcation. And I'm not going to say anything about this in detail, only to say that it's a trick, in effect, that they're separating one electron to power the endergonic reduction of ferredoxin by coupling it to an energy releasing step here, which is an exothermic reduction of a heterodisulfide to a thiol. So hydrogen reacts with CO2 to release methane as a gas, and this goes on and on and on in circles, in cycles, if you like. The only product is methane, which is released as a waste product. So the only thing which is conserved from this reaction is the proton gradient across the membrane. So as this methyl group is transferred from one cofactor to another one, there's enough energy released to pump a proton across that membrane. And so the question is, well, what are they doing with it? And the answer is, well, they're using this membrane protein here called an energy converting hydrogenase to power the reduction of ferredoxin and CO2. So this is the key protein in my mind at the origin of life. What this is doing is using a natural, geologically sustained proton gradient to power it's taking electrons from hydrogen, putting them onto ferredoxin and then onto CO2. In other words, this protein is powering growth. This protein is converting gases into organic molecules at the origin of life. It's an iron sulfur protein that sits in the membrane that reduces this other iron sulfur protein that I've already mentioned, the ferredoxin, and it's using what could be a natural proton gradient in a geochemical environment like a hydrothermal vent to drive that reaction. How is it actually doing it? Well, we don't know for sure, and I'll put some suggestions forward in a moment. But the first question before we come to that is, can it really do it based in a geological environment? If we work out the actual energies involved, the answer is, well, actually, yes, it can. So this was a mathematical model where we have a protocell set inside a barrier here. This is not intended to be realistic. This is intended to be the basis for a mathematical model where half of this surface is exposed to acidic fluids and half is exposed to alkaline fluids. And we can specify the permeability of the membrane itself to proteins, to protons, and we have proteins which are embedded in the membrane, and we can work out what's the flux rate through these. So these are all based on modern values. And we use the standard equations of electrochemistry to calculate effectively the rate at which protons will flow into this protocell and the rate at which they will be passively removed from the protocell by this alkaline hydrothermal flow. So protons will react with hydroxide ions and be dissipated. And so proton flux, in principle, can continue. It can only continue if we do not have a modern membrane. The problem with a modern membrane is it's impermeable to protons, or very close to impermeable to protons. So protons can come rushing in, but they can't leave again because the membrane's impermeable to them and there are no pumps. And that means the pH gradient between the inside and the outside collapses in our model within a second or so. Even a 5 pH units difference between the outside and the inside will collapse immediately because protons can come in through the proteins, but they can't leave again. But we see a very different picture if we have a very, if you like, leaky membrane, something like a fatty acid membrane, bilayer membrane, which is very leaky to protons. And here, this is what you've seen with a modern membrane. You lose all power straight away. But if you have a very leaky membrane just made of fatty acids, the protons can come in, but they can leave passively, taken away by this hydrothermal flow. And that means that you have enough power in this system to power everything in modern biology. That includes the energy converting hydrogenase that I've mentioned, and it includes the ATP synthase. So in principle, hydrothermal vents can power the reaction of CO2 and hydrogen to drive all of metabolism as we know it in cells like methanogens. So, but it requires a leaky membrane. It will not work with a modern membrane. And this potentially can explain the divergence between bacteria and archaea. This is a complicated slide. I apologize for that, but this is essentially what you've seen already. This is what I've just shown you without any pumps at all. This is the situation in methanogens that also you've seen. This is the electron bifurcation part here linked to a methyl transferase, which is the pump. And we still have an influx of protons coming through this energy converting hydrogenase. And bacteria have simply inverted that flow and are pumping protons out, and they've had to wire differently the electron bifurcation. So they're now fixing CO2 by a different route. It was forced on them by the change in flux through the energy converting hydrogenase. This is a hypothesis, but is testable through phylogenetics. I keep mentioning this energy converting hydrogenase. This was the atomic structure, atomic resolution structure from a couple of years ago. And it's in fact the core of the complex 1 that I showed you earlier on. So we still have this in our own mitochondria powering our own energy metabolism. This is not some weird enzyme only found in methanogens. It's inside you as well. And it still has these iron sulfur clusters there in the matrix arm here. And it can work as all these pumps can. It can work in either direction. It can pump protons out, or it can use protons as they come in to drive work. And that's what I think is happening here. This is, I showed you this very briefly already. We have an influx of protons coming in. We have these iron sulfur clusters right next to the membrane here. And what's happening is electrons are being stripped from hydrogen with a reduction potential of minus 300 millivolts under characteristic conditions for modern methanogens growing, so 10 pascals partial pressure of hydrogen. And it will pass them on to ferredoxin, which is mostly reduced in cells and therefore has a reduction potential of minus 500 millivolts. So this reaction should not happen. There's a 200 millivolt difference there. It should not happen spontaneously. It's being driven by the proton gradient, and at least two of these iron sulfur clusters, their activity is linked to pH. Their reduction potential is linked to pH in the same way as hydrogen and CO2 that I mentioned already. What that means is that they become, if a proton comes in and binds to these iron sulfur clusters here, that means it's easier to pass electrons onto it. So hydrogen can pass electrons on to these iron sulfur clusters, but they're not reducing enough to reduce ferredoxin. Then the channel closes, the two protons detach, and this becomes more reducing. It becomes more reactive and will now force its electrons onto ferredoxin. And so we've got a kind of redox switch here, which has got very simple components. It's got just iron sulfur clusters, a pH gradient, and gases like hydrogen and CO2. So it's a very simple system that we're considering. And so the question is, can this work in a purely abiotic, prebiotic environment in these hydrothermal vents? And we can reduce this to two possibilities, one of which is that hydrogen gives up its electrons onto the barrier. So we have the two phases here that I mentioned, and the electrons cross the barrier to reduce CO2 to organic molecules. And if that continues and these organic molecules build up into eventually protocells, then they would form on this side of the barrier. The alternative is that the protons cross the barrier, and CO2 is crossing the barrier, and this reduction is taking place right on the inside of the alkaline side, so the hydrogen is giving up its electrons here. That would give something which is more consistent with modern biology as a topology, at least. These are questions we've been testing, and I'm aware I've only got 5 or 10 minutes left, so I will push ahead. But we've been testing these questions for the best part of 10 years now. This was the first prototype reactors we built. We've been looking at microfluidic chips more recently. We have had some success in reducing CO2 to form carbon monoxide here, for example, formate, formaldehyde. I have to say we've not had very good reproducible results. I'm not really happy with our results. A former student of mine, though, has recently demonstrated, I think conclusively, that the hydrogen gives up its electrons on this side of the barrier, and probably the electrons are crossing the barrier, but it's not clear exactly what's crossing the barrier. But certainly the CO2 is being reduced by electrons from hydrogen, and it's picking up the protons from the acid side. And this is done with NMR, with 13C NMR and proton NMR. And the difference in coupling here is showing that the CO2 definitely is the 13 CO2 is providing the organic material in formate, and the protons for sure are coming from the acid medium here. But we still have this question: what is crossing the barrier? And to have this topology consistent with modern cells, which I was showing you very early on, this is the same kind of pore structure. We have protons crossing a barrier to power the reaction between CO2 and hydrogen to form organic molecules. If these organic molecules are formed here and form protocells, then we have a topology which is consistent, and that predicts that protons will be crossing this barrier. So we've done some work looking in microfluidic chips. This is just showing pH gradients with laminar flow in these chips. And if we precipitate a barrier, it's very noticeable here that the barrier is slightly yellow. It's acidic. So it looks as if protons cross that barrier more readily, and we confirmed that. This is the alkaline channel here with pH 11 from the pH indicator, and this is a diffuse barrier inside the acidic channel, and you can see that it's acidic right up to the very edge. We did some stop flow experiments and showed that that protons are crossing this barrier about 2 million times faster than hydroxide ions are going in the opposite direction. And what that means is they can be very steep pH units, 5 pH units across 70 micrometers, or possibly as steep as 3 pH units across single nanocrystals of iron sulfide of about 25 nanometers in diameter. So we've tried to model this, and we've started out, this is the same model that I was showing you before. We have a protocell wedged in a barrier, and we're imagining, and we can test these, that there are interactions between iron sulfur minerals forming as minerals and organics in the system, such as amino acids, and that this would be a proto energy converting hydrogenase, basically some iron sulfur clusters or crystals collated by monomeric amino acids. That would reduce CO2 to form organics. And as I showed you, they're likely to be amino acids, most of all, and fatty acids. We assume the fatty acids would go to the membrane and that the amino acids would interact with iron sulfur minerals and make it more likely that some of them would end up in the membrane, promoting CO2 fixation. So this makes some specific predictions that we can test. One of them is that these vesicles would have to stand up to extremes of pH between at least 9 or 10 or 11, and acidic ocean fluids of more like 6 or 7. The literature says that this shouldn't happen, that vesicles are not stable across these wide pH ranges that we would expect to see in alkaline hydrothermal vents between pH 5 and 12, at temperatures of 50 to 100 degrees centigrade, at modern ocean salinity, and in the presence of divalent cations like magnesium and calcium. In fact, though, this has changed a lot in the last few years. Tom McCollum showed 20 years ago that, that hydrothermal Fischer-Tropsch type synthesis under these kind of conditions can produce quite long chain hydrocarbons. So these are hydrocarbons with 15, 16, 17 carbons in the chain. And these are alkanols and alkanoic acids, so carboxylic acids and fatty alcohols with 10, 11, 12 carbons in those chains as well. And we have done some work, I won't go into detail on this, but we have shown a year or two ago that if we use these mixtures of the fatty alcohols and fatty acids that were produced by Tom McCollum, that they form stable vesicles with a bilayer membrane around them under extreme conditions, alkaline hydrothermal conditions, pH 12, 70 degrees centigrade, modern ocean salinity. I see I better come to an end in a few minutes. We're also able to show that interactions between amino acids and iron sulfur minerals will produce exactly the same types of cluster that we see in modern cells, so that in ferredoxin, so 4Fe, 4S clusters, we get them in forming spontaneously under exactly these conditions. We've been thinking about what would happen next. This is the model that you've seen already. This is modern metabolism, roughly as we would expect it to split up and to make nucleotides. I won't describe the modeling, but essentially it predicts that we should be able to make nucleotides under these conditions. And I would like to show you some experimental work showing that this is pyrimidine synthesis following these pathways, and we are able to make uracil this way, and much better under conditions that approximate to hydrothermal conditions. So 90 degrees centigrade, 100 bars pressure, mildly alkaline pH, and modern ocean salinity, we get 7 to 16 times the yield that we did under less optimal conditions, which is to say standard pressure, for example, and pure water and so on. We're also able to show, and this is more or less the final slide now, we're also able to show that ATP can be formed under these conditions, or at least ADP will be phosphorylated preferentially by acetyl phosphate under these conditions, exactly the same as happens in modern cells. It's acetyl phosphate specifically that does this, and we get a yield of more than 15%. And this is ATP being formed here, but it doesn't work with the other bases. We get nothing with UTP or CTP or GTP or ITP. So there's something favored about the adenosine ring, probably about this amino group in the adenosine ring, which is potentially why ATP is still the universal energy currency. It was favored even under prebiotic conditions. So this is the very final slide. How does chemistry come alive? I would say it's happening when a focused, sustained environmental disequilibrium of hydrogen, CO2, and pH across a porous structure that lowers kinetic barriers to reaction continuously forms organics that bind and self-organize into protocells, within which protometabolism generates catalytic nucleotides that can promote protocell growth through positive feedbacks. That was the model that I didn't really have time to go into. These crowded conditions inside protocells should favor physical interactions between nucleotides and amino acids, and RNA emerging in this protocell environment would be selected then, presumably, if it promotes protocell growth, and that gives genetic information meaning from the very beginning. Most of the experimental work I've shown you today has been done by my group, and they are brave, I think, in going into these questions, and we're starting to make some good progress. But I'm going to stop now. I need to say thank you to the funders as well, and thank you to you for listening. I'm sorry if I've overrun slightly. I will stop sharing now.
Thank you kindly, Professor Lane, for your interesting presentation. We'd like to now invite questions from the audience, from the participants of the summer school. May I please suggest that you raise your hand, and when you are called upon, can you please introduce yourself? Just say your name as well as where you're from. So we've got a question from Niket. Niket, would you like to unmute yourself, please?
Yes, sir.
Go ahead, please.
Yes, sir. My structure is like, can you a little bit more explain about that structure of complex mammalian one, and also like this another question is like H plus crosses more fastly like 100 times you told around than hydroxyl ions. So can it be related to like physics also, like mobility of electron is much higher than holes, so it gives Hall effect potential difference, and that's why means so many phenomena are seen in the semiconductor physics also. So can this biology of H plus and OH minus be related to that electron hole mobility also in some way, in energy generation of cells, and then like electron holes in these materials?
I struggle to quite catch all of that, but I think that what I've been showing in terms of the electron transfer and proton movement and so on is very much mainstream physics, but it's not really been integrated very well into biology. It should generate fields as well, and there should be quantum tunneling of electrons through these minerals. Very few people have worked on that, to my knowledge. My feeling is there's the nice thing about this is it is analogous to the way that cells work, and we see roughly similar things in geological environments. And I think that it's physics really which is going to come to the rescue, if you like, here, in terms of what the biology is doing is showing the overall topology and the analogy between biological systems and geological systems. And what I think the physics will show is the way in which electron movements and proton movements are linked and will generate fields, and the way in which those fields will effectively give unity to entities like protocells under these conditions.
Yes, sir. So like energy transfer mechanism also differs from organisms, and like so many differences are also seen, like cell walls are present in plant cell, but there is only cell membrane in animal cell. So
but all
like,
cells have a cell membrane. Whether they have a wall or not is almost not relevant to actual membrane bioenergetics. Of course it is relevant, but it's not the main factor.
Yes, sir. So okay, thank you, sir.
And thank you, Prof. Lane. We have another question by Jitendra. Jitendra, can you please unmute yourself?
Hello, Professor Nikhil. This is Jitendra Huda. I'm a teacher in biological sciences in international school in India. And I was interested in asking a question about the specific spin which we have in the electrons and the protons, which is very mandatory for regulation of the proton field or the electron field, for the energy reactions which are taking place inside all the cells, even the cells which you have talked about, the prokaryotic cells, the archaea and the eubacteria kind of cells, as well as the leuca kind of cells. So over there, the spin of the electron plays a very vital role. So I think somewhere or the other, if you can shed more light on the spin of the electrons, that will be a great advantage. in this session?
Yes, I mean, my understanding is the spin of electrons today depends on the chirality of the amino acids that make up the proteins. And so in the respiratory system, yes, they are in the same spin state. And they lose that spin state when they're passed on to oxygen. And so this can be measured and has been measured, to my knowledge, by Luca Turin, among others. It releases radio waves when they lose their spin. Now, the prebiotic environment I'm talking about does not have anything equivalent to chiral amino acids. They are almost certainly brought about by metabolism itself and by enzymes. As soon as you have enzymes, then you have a necessary chirality, because enzymes function by a hand-in-glove type mechanism. There are geological and physical ways of producing chiral amino acids, but very rarely do they do so to the level that we see in biology, basically 100% chirality one way or another way. So how important electron spin is in these processes, I don't know. My feeling is it's not of fundamental importance, but it may be of importance in ways that I have not begun to imagine yet. I hope that helps in some way.
Yep, probably I have a bit of answer regarding this, that there are certain reactions which are not possible to occur unless and until the electrons and the protons have a specific spin. And the amount of spin should have been maintained in a specific level, so that
Which kind of reactions do you have in
the
mind?
Leuca cells.
Which reactions do you have in mind?
If I talk about, I'm not very sure about the reactions as of right now, but I read it somewhere in an article that spin matters a lot.
Well,
so I don't exactly
the actual
remember
experiments suggest, and there have been quite a few now, that it is possible to reduce CO2 using hydrogen, using minerals in geological systems, minerals like greigite, for example, mckinowite and green rust, so oxyhydroxide minerals. I don't know if anybody has ever looked at the spin state of electrons or protons in these systems, but the fact is that geological systems will reduce CO2 to organic molecules. And so the evidence that we have is that it works in a prebiotic system.
Okay, thank you. Is that clear, Jitendra? Thank you, Professor Ling. Maybe could I ask you a question?
Of course, yes.
Chair? So from your presentation, it appears to be a highly complex process, right, that's involved here.
This is
so
a small part of the complexity.
Yeah, nonetheless, could you suggest how would such a level of complexity come about in the first place?
Well, the system that I've been talking about, essentially the simplest form of it is a type of hydrothermal vent where you have naturally occurring gradients of protons across naturally occurring barriers. We know these systems today. We have fossil evidence of these systems. We know all that's required is a wet rocky planet. We see evidence for this same process of serpentinization historically on Mars, currently on moons like Enceladus and Europa. And wet rocky planets, we know there are probably tens of billions of them in the Milky Way alone, based on the search for exoplanets. So wet rocky planets are common. They will give rise to this geological process of serpentinization. And that will produce these kind of hydrothermal vents that I'm talking about. They have a natural structure. They're rich in hydrogen gas. They have natural barriers. They're almost certainly in an anoxic planet. Almost certainly there will be ferrous iron available. And so there will be the catalysts and the barriers are naturally available. So what you want to read into that, they would be very common, these conditions, and they're naturally compartmentalized and structured, and I think should be driving the way that cell structure formed from the very beginning. And it explains why all cells are chemiosmotic. All the cells that we know on Earth basically have this same structure. it stumped me for a long time because I was thinking about ATP synthesis and the ATP synthase, which is plainly a product of genes and natural selection. So it's not possible to explain the ATP synthase in a prebiotic environment. But if we think about CO2 fixation and growth as the primary issue, then it becomes much more easy to understand how you can go from a relatively simple but structured system present on any planet, any wet rocky planet, to a more complex system that will evolve complexity over time.
Okay, thank you, sir. We have the next question by Vasudev Rao. Vasudev Rao, can you please unmute yourself?
Hello, is my voice clear?
Yeah, very clear.
Yeah.
Okay, Nick, thank you very much. Very nice lecture. My question is, how big is the gap between the organics that are formed in the vents with this process and the first living cell? How big is the gap?
It's a huge gap. The organics that are formed, what has been done experimentally is to form carboxylic acids, so Krebs cycle intermediates, sugars from there, and amino acids. And my own group have been looking at nucleotides, and other groups are doing that as well at the moment. So these are the monomers, these are the building blocks. We have not succeeded in polymerizing them to make RNA or DNA or proteins in this context. And even when you have the polymers, there's been a long question in the field about where does information come from. So what this system does, in my view, is it provides a context where we have the monomers, which have been made already, which naturally self-assemble into systems that resemble cells. We can call them protocells, which have a bilayer membrane around them, which in principle can grow and make more monomers. There's a big gap to the formation of polymers like RNA, but if and when these form, it's conceptually possible to understand how they can influence the growth of these protocells. Another big problem, I think that potentially solves the problem of the origin of information in biology, because the information is coming into the context of growing protocells before RNA itself. If you put RNA first, then you have a problem with how does it gain function. If you put a naturally driven system first that already has the function of growing, then it's much easier to understand how information can either improve or interfere with growth. The step to molecular machines, I think, is a big step. How do you go from simple polymers to the kind of complexity of a ribosome? I think we're a long, long way from that level of understanding that level of complexity in biology. So the origin of molecular machines is still a big issue.
Thank you, Nick. Thank you for a wonderful lecture.
Thank you.
Thank you. Just a quick question about where do you think consciousness plays in this whole model?
Well, I personally see consciousness as a property of biological systems and not of the universe. So I imagine a lot of people here might disagree with that, but I would see it as a property of cells, not of nervous systems. And I personally see the origins of consciousness as linked to the origin of fields surrounding cells, providing a real-time feedback on the physiological status of cells, if you like. It links the components of cells in a way that nothing else does. So I see it as essentially electromagnetic fields on the surface of cells. The same proton gradients that I've been talking about across membranes are playing another role, which is linked to consciousness.
Thank you so much, Professor Nick Lane. It's been a pleasure having you on our summer school. Thank you so much for joining me with a round of applause for Professor Lane. Thank you so much.
Thank you.
Thank you.
I'm sorry I'm unable to stay to listen to the rest. I would very much like to, but I'm afraid I have to go to a meeting with the funders who've been paying for this work, and I can't miss that. I apologize. Thank you very much for the invitation.
Yeah, Professor Nick, myself, Devasis. So
hello.
I should congratulate you, and many thanks for your receiving the invitation and also for this superb lecture. And thank you so much.
Thank you very much. I appreciate that.